So for those people who have heard my previous talks, you will notice that we have to go back to some things I have already told. But today we will use all those data for a very different question to answer. And the broad question is basically, what's wrong with breed bees? And let me take you slowly through this talk then. The problem that I was confronted with when I started to get involved with bee research was that the Varroa mite is already more than 40 years with us. Most honeybees, and certainly not breed bees, are after 40 years still not resistant against the mite. It would be desirable that they were, but the problem is that we are different from the rest of the world. The USA, Canada, and Western Europe, they have this problem, but in other parts of the world, the Varroa problem has spectacularly disappeared. This is true in South Africa, where they have very well documented from the moment that the Varroa mite arrived on what happened. And in 5 to 7 years, natural selection had resulted in absolute resistance. And so the question is, why is there this difference? Why does it work in one country and not in another country? For a long time, I thought, well, South Africa, there are lots of wild bees, and the bees that are used by the beekeepers, they are very often bees that are collected as a swarm in nature, and so they have not done a lot of beekeeping on those bees. But if you look at Western Europe or North America, people do all sorts of things when they keep bees. And most of the things that beekeepers do is trying to prevent natural selection. I saw what the program was for a starter course in beekeeping in the Netherlands. I could summarize the program by saying, the only thing you learn is how to prevent reproduction in bees, because you have to remove young queen cells, and you put small colonies together to make one, and you try to prevent swarming. And so there are lots of, and swarming is the way honeybees reproduce. So beekeepers have a tendency to work against natural selection, and so I thought that's the explanation of this problem. It cannot be helped, because that's what we do. And then I continued to read literature, and even rereading literature I had already seen before. And then I realized that this could not be the whole explanation, and that the problem was not so trivial as I thought. And that's what this talk is about. I am an evolutionary biologist, and evolutionary biologists use jargon. And that makes it a little bit easier to talk. But I'm going to explain a few words, and then that's tedious, but we do that first, and then I can use those words later. So one of the things we are going to talk about is a panmictic population. And a panmictic population is a population which is well mixed, meaning that animals from all over the place have the same chance to mate with each other. And you could imagine that in the 19th century, the chance that a French lady would marry an English man was rather small, because they were isolated. But nowadays, with internet and all the travel possibilities we have, the chance has increased. So people have become more panmictic. They mix better. And then polyandrous, that means that a female mates with a large number of males. And now we go to some terms in genetics which are important. I will talk a lot about alleles. That word you really have to remember. And an allele is very simple. It's a variant of a gene. Those variants of genes, they can have different properties. One is they could be recessive. Most of the alleles of our genes are recessive. Means that you need 2 copies to see the effect of those genes. But they're rare, but they exist, dominant alleles. Those, if you have one, the effect is immediate. And when an individual has 2 copies of the same allele, we call it homozygous for that gene. And when it has different copies, we call it heterozygous. And then the most important term that we'll meet today, recombination. Recombination is a way in which animals can make new genotypes. So, because the alleles coming from the father and the alleles coming from the mother are mixed by a process in which the chromosome breaks into equal pieces, and then the broken parts are swapped between the original paternal and maternal chromosome. We'll see more of that. Now, here to explain, if you have blue eyes, you can be sure that you have 2 alleles for blue eyes, because the allele for blue eyes is recessive. So you have only blue eyes when you are homozygous for blue eyes. But brown eyes are on an allele which is dominant. That means that if you have brown eyes, it could be that you have 2 alleles for brown eyes, but it could also be that you have only 1 allele for brown eyes. You can't see the difference from the eye color. And so it's possible that a couple that both have brown eyes will have a child with blue eyes. A couple that both have blue eyes cannot get a child with brown eyes. Now here you see this recombination. Originally, when a queen is going to produce an egg, all our cells are first diploid, so they have 2 different copies of all the genes, one coming from her father and one coming from her mother. Those then come together, they are arranged next to each other, and then the chromosome breaks, and the paternal and the maternal chromosome swap these parts. And that means that the children coming from some egg are different from the genetic composition of the mother, because the genes on the chromosomes are mixed. You see here the end result. Now we talk about Varroa resistance a little bit, and there has been an awful lot of research on resistant Varroa bees, populations that are resistant, and in those populations what you find is there are 2 important characteristics, or rather 3. The first is that bees that are resistant for Varroa, they have a high incidence of Varroa-specific hygienic behavior. They remove larvae from cells or pupae from cells on which there are reproducing Varroa mites. By doing so, they kill one of their sisters, but they also prevent the reproduction of the Varroa mite. So if they do that often enough, the Varroa mite cannot increase in number. And then, which is important for bees that are wintering when there's no brood, then all the Varroa mites, they sit on the bees and they suck hemolymph from those bees, and that's bad, because if you want to survive the winter as a wintering bee, and when Varroa mites suck your blood, then there is a good chance that you will not survive the winter. Now, bees that are resistant, they have the ability to brush off a Varroa mite. That's rather difficult, because the Varroa mites are very fast and they cling to their body, so you have to be very clever to try to get rid of them. And the Varroa mites also like to sit on places where the bee cannot reach with its legs to brush it off. But bees that have this property, they have a little dance that invites other bees to help her getting rid of the Varroa mite. So then there is an other bee that comes from the outside to brush off the Varroa mite. And when it falls, then the bees try to grab it with their mandibles and to kill it. So those two behaviors are essential for resistance against Varroa mites. And then you must know that bees which are sensitive to Varroa, then they have a growing Varroa population, and in the end, maybe after 1 or 2 years, the colony dies. And it's not because there are too many Varroa mites, but because the Varroa mites have brought viruses with them, and it is the virus that eventually kills the colony. So you would expect that bees which are resistant against Varroa mites are also resistant against those viruses. Because in Europe, resistant bees, they are not very common, scientists have decided to do experiments in which they set aside a large number of colonies and then stopped treating those colonies against Varroa to give natural selection a chance and then to see what happens. Most of the experiments, there are in 4 places or 5 places they did those experiments, 2 places in France, 1 in Norway, 1 in Sweden and 1 in the Netherlands. Most of those experiments have a very bad design and you cannot trust the results because of that. But there is one very elegant experiment, and that experiment helped me to realize what the real problem is. And that experiment was done by Ingmar Vries. Ingmar Vries decided to go to an island, Gotland, and to go to the southern tip of that island where there is a peninsula, and there he could have an isolated population of bees to make sure that natural selection could do its work and that there was no influence from other beekeepers. And so here on the left you see this southern tip of Gotland, and on the right you see Ingmar Vries. What comes out of his research is that alleles that are necessary for getting resistance, that those alleles have disappeared from the Swedish population. And I'm going to explain how that worked. And then there's a second thing which made me trigger to me the fact that I thought, it's very strange. So we have 40 years of research into Varroa and the viruses they bring. And in those 40 years, in Europe, there are only 3 records of bees that are resistant against deformed wing virus. That is very little. So I think alleles for resistance against the deformed wing virus have also disappeared. So we have a mystery. Now let's look at more detail at the experiment of Ingmar Vries. He started with 150 colonies, and after 2 years there were only 13 alive. So most of the colonies died because Varroa made them, and the viruses made them die. Now the interesting thing is that he really thought how to set up this experiment. So what he did, before he started to bring these 150 colonies to Gotland, he went everywhere in Sweden to collect colonies. And he made sure that he selected those colonies so that they represented all the different bee varieties which are kept by beekeepers in Sweden. And so he had Carnica bees and Ligustica bees and Buckfast bees in his experiment. And all those bees, they are breed bees. The interesting thing is, Those 13 colonies that survived, Locke, who was a colleague, studied those surviving colonies and checked whether those colonies had this hygienic behavior. And they didn't have it. So after a lot of selection, what remains does not have this behavior, and this behavior is important in surviving from the Varroa. And nobody, myself also not, didn't realize what that means. And what it means is that not only those 13 colonies did not have Varroa-specific hygienic behavior, but all the colonies that died also didn't have it. So he had sampled 150 colonies from all over Sweden, which is a large area, and it was really a good representation of the Swedish bees. And now we know, because he had taken care to do that, that there were obviously very little Varroa-specific hygienic genes in the Swedish population. They were not there. Otherwise they would have been in the sample, and otherwise they would have survived the experiment. And so the question is, where have these genes gone? Because most bee populations, maybe they don't have a lot of resistant genes, but they have some. So again, this mystery, the mystery of the alleles that have disappeared. Now we can look at the surviving colonies and some of their properties, because after all, they survived. Now, surviving colonies, they were very small, and they swarmed often. Now, swarming often, that is not good for Varroa, and certainly not in Sweden, because Sweden has a very short season. So you have an interruption of the reproduction, and then it's difficult for the Varroa population to build up again. So that was a helpful character, and obviously he had selected on swarming behavior. They also found in doing the genetics of those bees that they had become very homozygous, which is not good for bees, as you might know. So there were all these effects of strong inbreeding. There was one thing which was interesting, that if they opened the cells to see whether they were infected by Varroa, they discovered a lot of cells in which there was a female Varroa mite, but the female Varroa mite had not reproduced, it had not laid eggs. And that's interesting. That is what's called a brood effect. And a possible explanation is that the mites wait before they start to lay eggs from a signal of the bee, that the bee is going to pupate. And the bee has juvenile hormone, and the concentration of that juvenile hormone peaks when it's going to pupate. And people think that the mites use that signal to start reproducing. And it seems possible that in these bees, that signal was not strong enough to tell the mites that reproduction could start. And so they sat there and waited and did not reproduce. So that was at least an explanation for the fact that those 13 colonies survived. And the other thing that was very nice is that this is one of the 3 studies in which resistance against the deformed wing virus was discovered. The bees survived because they swarmed more often, because they had this brood effect hampering the reproduction of the mites, and because they were not so susceptible to the deformed wing virus. But the bees were not resistant. When those 13 colonies were there, they made sure that they would always, each year, breed queens from those colonies, and those queens have been spread throughout Europe and been used in all sorts of research and projects, because people thought, well, maybe that can help other people to get resistant bees. But that was not the case. As soon as you move those bees out of Sweden, then they are as vulnerable to Varroa, and they die as quickly as non-resistant bees. We have the problem, we know. We are looking for alleles which are no longer there. And now we have to try to find an explanation. What happened to those alleles? My hypothesis is the following. Selecting artificially for traits that beekeepers like, as low aggression, high productivity, and low swarm propensity, that has exhausted the genetic variation of breed bees in Europe and North America, and not elsewhere, because they do not do an awful lot of selection elsewhere. That's an hypothesis. Now, to test the hypothesis, then we have to come up with first with a sort of a mechanism how that would work. Why do you exhaust the genetic variation? Now, and when I discuss with breed breeders about this question, they always get a little bit angry with me, and they say, we are very good breed breeders, and we take care of the genetic diversity. We use a lot of different selection lines, and we test whether there is no homozygosity, and so we are quite good at what we do. And what's more, they are also successful. Obviously, in most of the breed bees, there is enough genetic variation to select and arrive at the properties that you're looking for. If you want to increase the honey production, or you want to decrease the propensity to swarm, you see results after a few years. The bees still respond to selection. And so then the breed breeders tell me, your hypothesis is rubbish. And then I say, no, I don't agree, I'll explain you why. And I'm going to explain you why. In the face of sufficient genetic variation to select for certain properties, there is still a loss of resistance alleles. That's what my claim is. And so we have to find the mechanisms for how these alleles were lost. Now, there are two possible mechanisms. One I have called the loss of rare alleles. And I will explain you in a while how that works. And the other is, and there's a lot of literature on that, that if you select on a behavioral trait, then you get homozygous regions in the genome. I think it's always good to have sort of a cliffhanger in the middle of a talk that keeps people interested. So we're going to do an intermezzo first. And the intermezzo is that I want to tell you about how bees, how healthy bees in nature take care of pathogens and illnesses. Because if we understand that, we can also understand what has disappeared. Now, honeybees, they are, by the way they live, they're very vulnerable to infectious disease. Honeybees have only one generation per year, and so they have basically small populations, because for the population size you can only count the queens and not the workers. So a small population of reproductive animals. And bacteria and viruses are very different. They have many generations per day even. They can divide every 20 minutes under optimal conditions. So they have many generations, and they can occur in extremely large numbers. And every time a new bacteria or a new virus is made, there is the possibility of an error in copying the DNA, and then you have a mutant. And most of those mutants are rubbish, and they die out. But by chance, there could be mutants which are more aggressive and cause more illness. So there are new virulent variants. Now and then bees keep the brood nest around 35 degrees, and that's the best temperature to breed bacteria and viruses. A bee colony is very suitable for bacteria and viruses, and bees are always in close contact. So thousands of bees touch each other the whole day, and they exchange food, and they preen each other, and so they exchange also pathogens and viruses very easily. So that's the reason why bees are vulnerable. But bees have a lot also in their biology to resist. They prevent illnesses because they use natural antibiotics. These antibiotics are in the propolis and are derived from plant resins, but also pollen and honey contain lots of antibiotics. So they have a whole pharmacy in their food. And then they suppress illnesses. And the example we have been talking about is that they could have this hygienic behavior. There are also hygienic behaviors against microsporidia and bacteria. And the last one, and that's the one we are going to talk about for the rest of the talk, is that they, like they have also, each individual has an internal immune system. Let's look at that immune system more closely. We come up with a lot of characters that first I thought, well, yes, of course, bees are like that, we know. And it took me a lot of time to realize that all those characters, in fact, help to protect bees against illnesses. And the first one is this recombination rate, where pieces of chromosome are swapped between the maternal and the paternal chromosome. The bees do that at a rate which is the highest in biology, the highest in in the animal kingdom, and also higher than any plant. Because all organisms that reproduce sexually, they have that recombination. And so bees are really extremely exceptional in that they have this enormous high recombination rate. And when you read that, you have to think, why would that be? And we'll talk a little bit more about that in a while. And then bees have this well-mixed population, which I will show is important for this story. And then you have, and that you can understand, queens that mate with a large number of males, make that the workers in a colony are groups with different fathers. And so some of the fathers may have supplied resistance against an illness, whereas other fathers didn't, which means that the risk is spread and not all workers will die when there is an infection. And then yesterday I talked about the departure of the mother queen, which means that the genetics of the remaining colony where the daughter becomes queen changes dramatically, which means that there is a chance that the bacteria and the viruses living in that colony and were adapted against the genetic composition that goes with the old queen, that they all of a sudden are confronted with all sorts of new weapons that the bees have because the genetics have changed. And then finally, and I will not talk in this today about how sexual selection on the drones works, but I could say very brief, if you have a colony of bees that produces 20,000 drones in a season, which large colonies are able to do, then of all those drones, I calculated that there are only 2 who succeed in passing their genes on to the next generation, which means that the majority of drones has no chance to reproduce. So there is enormously strong competition between drones, and only the very best healthy ones which are fast and agile, succeed in mating with a queen. And because the queen mates with a large number of drones, then again, it's still a question if the semen of a certain drone will be used to make the next queen. It's a sort of lottery. So it's not easy to be a drone, I would say. Now, back to the high recombination rate. We cannot study the evolution of recombination rate directly, so you have to make a model. And these two very clever mathematicians, they built an evolutionary model in which they expose an organism that has a small population size and a generation time which is long, and they expose that to virulent bacteria, and then they look what happens if you let that system evolve in time, what happens to the recombination rate. And what they could do with their model is that they could tweak the virulence of the bacteria. And the outcome is a recombination rate. And if they make the virulence of the bacteria low, then the recombination rate that comes out is rather low. And if the bacteria become very virulent, then the recombination rate that comes out is high. So that's a proof, a theoretical proof, for the fact that the high recombination rate is an adaptation against pathogens. And so only strong selection by pathogens can result in this extremely high recombination rate. Well, we thought already that that was so because of all the risks that bees have living together in a warm place. Now, what's recombination rate doing? It creates new genotypes because it's mixing the alleles on the chromosomes. And so it may enhance, and it can, by doing so, it can bring together favorable alleles that sit on different genes. Because sometimes genes collaborate in attacking pathogens, and then it's handy if they are together and close to each other. If there's more recombination, then the probability that you end up with a good combination, of course, increases. But the problem is that if you continue to increase the recombination rate, then the probability that you play good combinations apart is also increasing. Which means that there is an optimum value for the recombination rate. And that's what the model I just told about has calculated. So natural selection optimizes recombination rate. Now the next step. Now we have to understand why it's so important that the bees mate in nature. The free mating bees in a large population, they are better capable of evolving resistance against pathogens. That's what I'm going to show. First of all, we have to prove that bees have such a population structure, which is not so easy. It started, this research, when the brothers Rutner in Switzerland looked at where drones of different colonies go to. So they painted drones of each colony in a different color, and then they went to a place where drones come together to wait for virgin queens, and then they would try to see which drones were there and where they came from. And in that way they were able to show that drones of one colony, they go to different congregation sites, and drones of many different colonies may visit the same drone congregation site. And that was better studied later with molecular data by Emmanuelle Baudry. What she did was, in an area in Germany, she mapped all the beehives and all the bee colonies that were not in hives, and then she collected bees from all the colonies, And then she did a DNA analysis, so she could recognize bees from colonies because they all differ in their genetics. And then she went to a big drone congregation area, and one day, she did that in one day, she was catching drones with a net. And she killed those drones, and she took their DNA, and then she could know from which colonies these drones came. And in that way, she was able to show that on one day, on one particular day, there were drones from 240 different bee colonies in one site. So that shows that the drones, at least, are well mixed. And then Astrid Jensen did another experiment up in Scotland in the valley, and she also worked with DNA samples from colonies and from queens and drones, and she could show, so she let the queens mate freely up in the air, and because she knew the fingerprint of all the colonies, she then could look at where the drones came from that the queen had mated with. And that was interesting because she could show that some drones and queens lived 15 kilometers apart. Most matings were with drones that came from a shorter distance. But some 10% of the matings was up to 15 kilometers apart, which means that a queen recruits males from an area of about 250 square kilometers. So that is really a huge area. Together, these three studies, they show that bees really have this panmictic population. So now we are moving on. There's always one thing that people object. They say, yes, but I know that the bees in this region, they are locally adapted. They produce a lot of workers late in summer, and that's because we have all the heathland around us, and the bees are adapted to that. They're different from the bees elsewhere. So how do you get local adaptation? And that's also what Annette Jensen's study shows. 90% of the matings comes from a short distance, which allows for local adaptation. But 10% comes from far away, and that means that for those 10%, you can find genetic variants which can come from very far. Well, this is a picture to show that all the drones that a queen mates with are genetically different, and that you get an awful lot of different genetic variants in the workers, which means genetic variation buffers the risk of being sensitive to illnesses. It is because the new queen is genetically very different from the old queen that all of a sudden the genetics of the hive are interrupted. Now we have, so we had 5 different things that all play a role, but I said there are 2 who are very important, and that's the panmictic population structure together with the extremely high recombination rate. Let's see how that works. So the panmictic population structure allows the recruitment of rare alleles. Some alleles are very rare, and maybe you cannot find them here, but maybe there are bees 10 kilometers further that have those alleles, and you want them all, so because you want them because you need them now, and so the mating structure allows recruitment of those alleles. And then the recombination rate comes in, because now you have the rare allele, and you need it because you have been attacked by a new illness, and then the recombination rate makes that the allele can very rapidly be combined with other alleles on other genes that you need also. And now we are finally in a position to judge what happens with breed bees, because breed bees must mate with other breed bees. And they cannot do that in a panmictic population because then you don't have any control about what happens. So breed bees go to mating stations. And there they are mating in a relatively small population of bees that have all been selected for the same characters and are already a little bit inbred. So, and that causes homozygosity of genes, and we'll talk about why that is wrong a little bit later. But it also causes the loss of rare alleles. And that is the most important message for today, and I'm going to explain how that works. And that is really the message for today, that you understand once you select that you might run into problems. Because when I talk to bee breeders, they always say, well, you can select, and I'm doing selection, and I've done it for 20 years, and it works very well. And they don't realize that selection is possible, but it comes at a price. And today I'm explaining what the price is. Now, first of all, we'll talk about this homozygosity. And for that, you have to know a little bit more about genetics. Because a bee breeder would say, I am selecting for Varroa resistance. But it's not so that there is one gene for Varroa resistance. What you are selecting for is a change in rather complicated behavior. Because a bee must recognize a cell that contains reproducing Varroa. It must be against the normal procedure. It must open, remove the cover from the cell to open it. And then something which is also runs a little bit against nature is then it has to grab a living sister and drag her out of her cell and kill her. So that's rather complicated behavior. And the same holds for improved honey production. Improved honey production means all sorts of things. It could affect the dances of the bees. It could affect their metabolic rate, the way they search, anything. So it's also very complicated behavior. Swarming, very complicated behavior. So those characters, they are based on many, many, many different genes. At least a couple of hundred. So if you start to select for such a property and you are selecting on a couple of hundred genes and you realize that most of the alleles in living animals are recessive alleles, then you realize that if the desired properties of selection, to reach those, you have to make a couple of hundred of genes homozygous. And the problem is that genes sit on a chromosome next to other genes. And very often, if you make one gene homozygous, then there is an effect, it's called a hitchhike effect, that you also make the sister gene homozygous. So you don't make only the genes involved in the selection homozygous, but also their neighbors. So you get stretches on the chromosomes which are homozygous. That's summarized in this slide. And now why it's wrong. I told you that high recombination rate is very important. And in a recombination, you swap different parts of the chromosome. But if genes are homozygous, then you swap the same thing. So it has no effect. So selecting for homozygosity results in blunting the weapon of recombination. So that's why we don't want it. Now this is not the worst. Now we get really to the most important part of the talk. And it's very simple mathematics. You must know that in a population of bees, all the bees have the same genes, but not all the bees carry the same alleles. And some alleles are rare, and other alleles are common. There's an example of black bees in Ireland, where they looked at the population and looked at the genetic diversity, and they found there were, of a particular gene, there were 37 different alleles. See? Some of those alleles are very common, some of them are rare. Now, you are going to select. Selection is taking a small sample out of a large population. If you take a small sample out of a large population, the probability that your sample contains common alleles is much larger than that you sample the rare alleles. The result of that is that the rare alleles disappear because you repeat the selection each generation, and you do all sorts of things that are also sample taking. For instance, you're a queen breeder. You take a frame, you watch, you see that there's an area with young first instar bee larvae, and so then you sample 20 of those larvae and you try to make queens of them. Then you have taken a sample from the whole colony, again losing rare alleles. And there are these other techniques. For instance, some people use single drone insemination, artificial insemination with one single drone. Now that's the smallest sample that you can take, and so it will result in a rapid loss of the rare alleles. So basically all what we do is, by keeping our bees as we want them, we are in a process of continuous sampling, getting rid of all those rare alleles. The next problem, of course, and you should have asked me, but you're not allowed to ask questions, and is, so what? Rare alleles. Why would we need rare alleles? And why are they rare in the first place? Why is it a problem? Now first I show this graph for people who are visually more prone to absorb things. You can see that the probability that you select in a sample a gene, it gets smaller when the allele is more rare. And that the more common it is, the higher the probability that you'll select it. Now we come to this question, why are rare alleles rare and why are common alleles common? Now first, the most easy part. Common alleles are common because they are favored by natural selection. So they must be useful under the circumstances, either for the bees or for the beekeeper. So that's one of the reasons that beekeepers can do this artificial selection, because they work with common alleles. Those are the alleles that they like anyway. And rare alleles, you have two types of rare alleles. You have alleles which have a negative impact on your fitness, and selection works against that. So those alleles, they disappear. They are not good for your health, so bad genes, they are selected away. So we are not considering those. But there is another group of rare alleles, and they're called neutral alleles. They do not affect your fitness. They're not bad, but at this moment they're not good either. What has happened is that because other alleles had been selected to become common, these alleles have become rare. And they're floating around in the population because at this moment they're not used. And if the world did not change the whole time, then we would not have to care about the rare alleles. But what happens is that the world is changing the whole time, and that a change comes. For instance, a new pathogen arrives or a new parasite arrives, and then all of a sudden a rare allele may be in need. And then a natural selection grabs it, and then it becomes a common allele again. And then another allele may drift away. Now you need a very large population to make sure that rare alleles do not disappear by chance. So that's why we have the panmictic population. A large population in which there are neutral alleles of genes that are floating around they're not selected against, they're not selected for. Those can be recruited when they are needed. That's the story. That's why it's very important that bees can mate in a natural way, going to the places where they can meet a lot of strangers and make sure that they can collect those alleles. So rare alleles are rare because currently they offer no advantage in natural selection. But as soon as the condition changed, some of those may be needed, and when the condition changed again, others of them may be needed. We can understand what went wrong with the Swedish bees, because they were breed bees. They had been selected for a long time. And so all the rare alleles had disappeared. And then Varroa came in, and the rare alleles were no longer there. So the population could not respond to those new enemies. And as I said before, breed bees cannot be part of a panmictic population. So when you mate them either by artificial insemination or by bringing them to a mating station, you are sampling. You take a small part of the population, so you're losing the rare alleles. Certainly you're losing those alleles if you do single drone insemination, but also when you're breeding queens from larvae of the same frame. And if you repeat those techniques in every generation, there's little hope of maintaining the genetic variation for resistance. And that's why breed bees have lost those. And that's why it's so important that we have wild bees. And the good thing is that black bees have been unpopular for a long time, so they have not been exposed that strongly to all these sampling problems. And so the black bee is a reservoir of resistant genes. Remember that. As I told you, beekeepers don't like my story. And they say, yeah, but we work with a large number of different selection lines to ensure the genetic variation, so we don't have that problem. It's our neighbor. And then I say, if you have selection lines, you're selecting in each line, and so you're sampling in each line, so you lose the rare allele from each line. Maybe the lines are a little bit genetically different, so you save some of the genetic variation, but you don't save the rare alleles. Oh, we didn't think about that. And the other thing they say is, well, we know that our bees are heterozygous. We don't have diploid drones, and we take care that our bees remain healthy. And then I say, and that's another problem that we should address, that heterozygosity is not a guarantee for the conservation of rare alleles. And I want you to see how that works. And now I'm going to do a rather devilish calculation. And what I do is I use this example that I mentioned of those Irish black bees that had 37 different alleles on one particular gene. Now suppose there were only 35. And now you take those bees and you're going to select for qualities that you like. And what happens is, because you're selecting, you lose 25 of the rare alleles. So there are only 10 alleles left over in your population. To make the reasoning easier, we say those 10 alleles which are still there, they're equally abundant. So they have the same chance of being chosen when you do a mating. Now, and then you can say that the chance of an egg, when it's fertilized, that it will be fertilized by semen with the same allele is only 10%. Which means that the heterozygosity is 90%. Now, if you look in the literature values for heterozygosity, 90%, that's a good high value. Then People say, oh, this is okay, 90% heterozygosity, wonderful. But you say that in the face of the fact that you've lost more than 71% of your genetic variation. So you see where the argument goes wrong? You think homozygosity saves you from everything, or heterozygosity saves you from everything, and you cannot see those rare alleles. You've lost them, but you've never seen them. You didn't know that they were there, but you've lost them nevertheless. So heterozygosity is not enough. And then there's another thing that is a personal irritation of myself. It is that if I talk to people who have exotic breed bees, then they're always very careful with the queens they have. They have these virgin queens, and they bring them to islands in the Netherlands where the mating stations are, and then they mate with the drones of the same type of bees, and that's how they continue to do their beekeeping. But what did they do with their drones? They just let them fly out in nature. And I think that is not a good idea. Because we know that keepers of breed bees have bees that have lost their rare alleles. But they release all their drones. So all other beekeepers, people that like black bees or people who like to keep bees in a more natural way with natural mating, they have this problem that there is an enormous number of drones with a low genetic variation. So even if you are keeping your bees in a more natural way, you lose genetic variation because your bees are mating with the drones of people that keep breed bees. Now, one other thing to show how dangerous it is very different from what I told you before, but it shows also that selection is difficult. And that's this spring, there was a paper by, already last spring already, Anthony Neerman and Dennis van Engelsdorp, and they did something interesting. They did an experiment on water collection by bees, but they repeated an experiment that had been done 50 years ago. And one of the results they were not looking for, they measured in their experiment how long bees live, the workers. And then they discovered that 50 years ago, the lifespan of the worker bees was 34 days, and nowadays it was only 17 days. I think it comes because they have selected for higher honey production, and when you do that, you select for a higher metabolic rate. And when you have a higher metabolic rate, you die sooner. And you have also a smaller chance to survive the winter. There are costs to the selection of things. Now, I wind up. So honeybees need a panmictic population structure to stay healthy. And the reason for it is that they can recruit the rare alleles. And the reason they have a high recombination rate is that they can make use of those rare alleles very quickly. And if the rare alleles are no longer there, then you have no selection for resistance against new pathogens. And so my idea is that we all keep black bees and that we will have a large naturally mating population of black bees that can act as a reservoir against new illnesses and new pathogens. And I'm absolutely sure that they will come. We will have more new parasites coming in, and we will have new variants of viruses, and there will be new strains of bacteria that will come in and surprise us, and we must make sure that our bees can develop resistance. And that's it.
So if I have understood your talk correctly, that there are always these rare alleles, but how can you be sure that there was the allele for Varroa resistance? You talked about it having been lost, but how do you know for sure that it was there in the first place?
Oh, because yeah, I showed that yesterday, I think. What we did was we looked at black bees, And there were people were starting a program on selection, and so they first had to know what is the resistance of those black bees that have not been under selection. And it turned out that they had very high frequencies of this Varroa sensitive behavior. So we know that in natural populations, and we have seen that the same applies to South Africa and to South America, where very rapidly those genes were recruited and very rapidly spread in the population. And Tom Seeley has shown the same for his bees in Arnold Forest. So normally it should be in populations that are mating freely. And it is by selection, therefore, that you got rid of them.
I think, Jacques, it's part of a culture issue, because people these days tend to just go and buy a queen. If they want a queen, they just go and buy one.
Yeah.
I think part of the issue is that raising your own queens isn't taught as much as perhaps it should be.
That is already very helpful, because in America there's one very large queen breeder in Hawaii, and most American beekeepers, they all obtain their queens from this one supplier, which of course is very dangerous. So breeding your own queens is much better. But also, when you collect the young larvae to rear queens, don't take them out of a single frame, take them from different colonies and different frames. It's more work, but it pays off.
The point about the black bees in England, I understand. You will be giving a different lecture maybe in New Zealand, where the native population is not black. And what about Australia? They have local bees there. You would be persuading them to hear from locals. Yeah?
I did refer to the bees in Arnold Forest. They're not black bees, they're a mixture of all sorts of bees. And even in those bees, evidently, there was enough of the resistant genes present to make the population under natural selection resistant. I guess that it's similar in Australia and in New Zealand.
So regarding your idea of focusing on the black bees, I am not so optimistic. I think it's in principle a good idea. So I give you an example. In Switzerland, there is a focus on breeding of the black bees.
Yeah.
And there's one state in Switzerland, it's actually a remote valley, Canton Glarus,
I know.
where it is legally forbidden to keep any bee other than the original black bee. But these bees are again bred, was selected for the needs of the beekeepers, and again the rare alleles will be lost.
Yeah.
So maybe, so would you actually propose, if you say were a dictator, to legally forbid any breeding station in the country?
No, it's not what I want. And I'll explain. I have noticed in discussing with the Swiss people that Swiss people are very different from Dutch people. Swiss people, they want perfection in all they do. They want perfection. And so what Swiss people think is that the only black bee, which is a good black bee, is a perfect 100% black bee. And that's part of the problem. And so they start then to breed those bees, and then they're breeding again, and then they're selecting again, and they're destroying the genetic variation of the black bee. My approach in the Netherlands is very different. I say, we have taken 150 years to destroy the black bees. So we may take maybe a century to restore the black bees. And we're going to do it very slowly by slowly increasing the frequency of black bee genes. And then it's possible to have, and that's the essential thing also of the way I think about these things, is that you must have breeding stations for professional beekeepers, because it's difficult to make a living as a professional beekeeper, and so you like to work efficiently, so you want highly productive bees, you want bees that are not so aggressive, etc., etc. And I think professional beekeepers should have the right to do that. But on the other hand, Most beekeepers are amateurs. They don't need to maximize their production. And so those could have bees that together with the wild bees that should be restored in our forests, then make a large panmictic population that can act as a sort of reservoir of rare alleles. And when the professional beekeepers need those genes because in their stocks they have exhausted the presence of the rare alleles, they can always go back to this natural population because in the reservoir they're still present. That's my ideal.